Hi students. So last night when we were on our little study session call, there were a couple of questions about the function of astrocytes. So I wanted to go over that a little bit. And so basically, astrocytes are very interesting cells. There's actually 6 to 12 times more astrocytes in the brain than there are neurons. So these are very numerous cells. Now, what's interesting is there's actually dozens of different kinds of astrocytes, and they look very different. And the theory is that they actually vary. They have very different. They have very different functions in the brain as well. And we're really just touching the surface of what we understand about astrocytes, but we're starting to realize that they are incredibly important cells in the brain. Now, it used to be that glial cells were just seen as the glue that holds the brain together. And astrocytes are a very important structural cell. Because they are so numerous, they provide the sort of the major parts of the structure that hold all the other cells in place. Um, so that is one of their important functions, that they are important structural cells. They're almost like the skeleton of the brain in a sense, though they're much softer than any skeleton that you could imagine. But their function goes way beyond this. And again, I think we're just touching the surface of what we know about astrocytes. I think in one of my prior videos I mentioned how when they started studying Einstein's brain, they realized that he had no differences in the number of neurons or the number of neuronal connections between anyone else's brain and his, but what was remarkable is that he had more astrocytes than most brains do. So, that sort of got people hypothesizing that maybe astrocytes have something to do with intelligence. And, there's been a lot of findings since that time that have made us really wonder about the function of astrocytes. And there's some people that think that astrocytes are connected together to each other through complex gap junctions. And so actually a complex network of gap junctions. And so basically there are, between each foot of, when two astrocytes come together, there are 6 little protein pores in between them, and that allows ions and other chemical signals to flow freely from one side of the gap junction to another. So this is really important because the astrocytes make up a whole separate network within the brain. Neurons are all connected together, but astrocytes are all connected together too. So there seems to be this chemical signaling that's going on parallel to the electrical signaling that's going on with the neurons. So again, we're just scratching the surface. No one really knows what this means and what kind of information is getting passed along, but we do know that it seems to be very important. So what else do astrocytes do? Well, the second thing that they do is they modulate synaptic function. And how do they modulate synaptic function? Well, if you remember, at the end of in the presynaptic membrane here, as a action potential is coming down the axon, it stimulates release of neurotransmitter from the presynaptic membrane into the synaptic cleft, right? So we have neurotransmitter being released into the synaptic cleft. And we talked about SSRIs and serotonin reuptake inhibitors, and we talked about the reuptake phenomenon, how important that is to the function of the synapse. Well, that reuptake is happening in the astrocytes. So the reuptake does not tend to, we used to think that it occurred in the presynaptic membrane itself, but now we know that it doesn't. It actually, the reuptake of serotonin and other large molecule neurotransmitters are taken up by the astrocytes and then recycled in the astrocytes and sent back into the neuron. So it's very important, and You know, what happens when you take an SSRI, you end up with more serotonin that's hanging out for longer periods of time in the synaptic cleft, and that tends to make the um, postsynaptic membrane a little bit more excitable, easier to excite because there's already a fairly high concentration of serotonin in the synaptic cleft. So by changing the amount of reuptake that's occurring, by making it more or less, the astrocytes can control how reactive the synapse is. So that's called synaptic modulation. So that's the second function. Now, another important function is the astrocytes have a very significant role in managing in storing glycogen. So glycogen storage and glycogenesis. And it actually has a role in providing other important nutrients to the neurons as well. So it is storing and releasing glycogen for neurons to use. Okay, so it has an important energy and nutrient storage function. Now, a fourth role is it has an important ion cleanup. Now, I know someone had asked last night about, I'm going to scoot this over for a second. Okay, so last night someone was asking me about what happens to the ion concentrations with, when there are multiple action potentials, and how does it get restored? Now, remember, because we have, the sodium-potassium pumps are constantly pumping sodium out of the cell and potassium into the cell, right? So we have lots of potassium in the cell, lots of sodium outside of the cell, and it is this difference in this movement of ions in and out of the cell that's providing a constant ion difference and voltage difference on the cell membrane. And then when we have a voltage-gated sodium ion that opens up to start an action potential, right, then we have a reversal of this flow momentarily. It really only lasts for a few seconds, where we have lots of sodium flowing into the cell, right? But this really only changes the concentration of sodium within a few molecules of the surface of the cell membrane. So it doesn't really have a significant impact on the concentration of ions in and out of the cell. And then, if you remember, after an action potential, the next thing that happens is to restore the positive charge on the outside of the cell. Potassium leaks out, right, through the slow potassium channels. And again, generally it's not enough to change the concentration of potassium on the outside of the cell, except for in the immediate vicinity of the cell membrane itself. Okay, so in the larger world of the extracellular fluid and the intracellular fluid, it's not making a huge difference. However, in the brain, where you have millions and millions of neurons crammed together very, very closely that are firing very, very frequently, sometimes you can end up with the electrolytes can begin to become imbalanced, and you can end up with a high concentration of potassium in the extracellular membrane. Now, this doesn't happen much in the peripheral nervous system because the nerves aren't very close together. This only happens in the brain when you have a lot of very active neurons firing together. Now, the astrocytes play an important ion cleanup role by soaking up the extra potassium ions. And because they're all connected together, these potassium ions can spread out through this huge network so they can be dissipated throughout the brain. And also, we're connected to, the astrocytes are very closely linked to capillaries. So if there is too much potassium to be dispersed throughout the network, it can just be dumped into the capillary network and be spread out. So the astrocytes play an important role of maintaining the ion balance so that we can have effective resting potentials. Okay, and then the fifth role here of the astrocytes is going to be scarring and repair. So if a neuron is destroyed or damaged, and then an astrocyte will sort of grow to fill in its spot. So it has an important role in scarring and repair as well. Okay, I hope that was helpful. Please let me know if you have any other questions, and I will talk to you soon.